Tregs. Regulatory normal mouse studies demonstrated mechanisms of T-cell tolerance. When CD4 T-cells were depleted of CD45 RCLAW or CD25+ cells, or following a 3-day thymectomy, the mice developed autoimmune diseases such as diabetes, thyroid issues, pernicious anemia, and diseases affecting ovaries and testis. Conversely, T-cell deficient mice without CD4 T-cells did not develop disease. This highlights the role of certain CD4 T-cell subsets in maintaining tolerance and preventing autoimmunity. Regulatory T-cells, Tregs. Tregs are a population of cells crucial for balancing inflammation and regulation. They play a key role in dampening down aggressive immune responses and are important in other biological contexts like tissue remodeling, recovery from traumatic injury, and regulating inflammatory processes in diseases like cardiovascular disease and neurodegenerative diseases. They represent an extrinsic or dominant way to regulate the body's immune system, working across multiple pathways, unlike therapies targeting single molecules like TNF-alpha. Removing Tregs leads to disease. If regulatory cells are depleted in a mouse, it develops chronic autoimmunity, indicating a naturally self-destructive potential within the immune system. A mouse thymectomized at 3 days develops gastritis or other autoimmune diseases. Putting effector cells into this mouse worsens the gastritis. However, transferring Tregs into this mouse prevents gastritis. This shows that Tregs are responsible for disease if absent and can dampen down disease if present. Transferring CD4 plus CD25 high Tregs suppresses autoimmunity. Tregs are typically FOXP3+. FOXP3 is the master regulator of Treg function stability. FOXP3 mutations induce aggressive autoimmune pathology affecting multiple organs, seen as IPEX in humans and scurfy in mice. FOXP3 controls different genes, upregulating molecules like CD25, GITR, and CTLA4, and downregulating IL-2. Most studied Tregs are FOXP3 Tregs. They are characterized by high levels of CD25 expression, the interleukin-2 receptor alpha chain. FOXP3 is the dominant transcription factor that imprints the regulatory phenotype on these cells. Tregs can be thymus-derived or peripherally generated. They constitute 1 to 10% of peripheral CD4 plus T cells in humans. They have the ability to suppress many different cell types. Tregs are important. Depletion of CD25 plus Tregs leads to spontaneous autoimmunity, underlining their importance in preventing autoimmune reactions. If humans lack these Tregs, they develop fatal autoimmunity. Tregs are important in operational tolerance in liver transplantation. Studies in liver transplant recipients showed differences in the number of FOXP3 plus Tregs in the blood of tolerant versus non-tolerant individuals. Treg balance is important in health and disease. Lack of Tregs is associated with autoimmune diseases and loss of tolerance to an allograft, organ transplant. In contrast, there is often Treg dominance in tumors or chronic infections. Tregs are also important at the fetal-maternal interface to tolerate the semi-allogenic fetus. Beyond immunosuppression, Tregs also adopt different phenotypes in different tissues and are responsible for tissue repair. For example, Tregs from the heart express factors for cardiac injury repair, and those from the nervous system secrete factors for neural tissue repair. They are being investigated in regenerative medicine. Tregs are involved in autoimmunity, allergy, metabolic inflammation, like cardiovascular disease, pregnancy, cancer, transplantation, and controlling host and commensal interactions, e.g., in the gut microbiota. Treg-focused therapies. A huge number of therapies are being developed to strengthen regulatory T cells. These include cell therapies and non-cell-based therapies. Non-cell-based therapies include low-dose Illinois-2 to promote Treg growth, drugs like rapamycin that stop effector cells from using Illinois-2, making more available for Tregs, TNF receptor agonists, and reverse vaccination approaches. Reverse vaccination delivers an antigen in a tolerogenic environment to generate regulatory cells, as opposed to using pro-inflammatory adjuvants which generate effector cells. Companies are exploring targeting resting dendritic cells with lipid nanoparticles carrying autoantigens or alloantigens to promote tolerance. Cell-based therapies involve generating populations of regulatory T cells for adoptive cell therapy. This is considered a potential next pillar of medicine alongside small molecules and biologics. Adoptive Treg therapy. Advantages of adoptive cell therapy include ability to comprehensively characterize cells phenotypically and functionally before infusion. Delivery of a tightly controlled dose of cells. Potential to select cells of known antigen specificity. Cells are responsive to the system they are in, have complex sensing and response mechanisms, can home to target sites, naturally or engineered, and act as a live drug deciding whether to regulate at a site. However, they require dynamic control as they are living. Suicide switches can be built in to eliminate cells if they become potentially dangerous. The general process for adoptive Treg therapy involves taking regulatory T cells from a person, blood or leukapheresis product. Isolating the cells using sorting methods like flow cytometry or magnetic beads, CD4 plus CD25 plus CD127 low phenotype is used as FOXP3 is intracellular. Expanding, invigorating, and potentially manipulating genetically changing the cells in vitro. Large amounts of Illinois-2, anti-CD3, and anti-CD28 microbeads are used for expansion, often with rapamycin to inhibit effector cell growth. Freezing and characterizing the expanded cells. Infusing them back into the patient. Adoptive Treg therapy was shown to work in early mouse experiments and in multiple mouse models of transplantation, inflammatory diseases, and autoimmunity. In the NOD mouse model of type 1 diabetes, Treg transfer can prevent and even reverse diabetes. Highly aggressive therapies like anti-CD3 or ATG can prevent disease, but Treg therapy is one of the few that can cure established diabetes in this model. Early human trials of polyclonal Tregs have shown safety and feasibility in multiple settings, including T1D. However, efficacy has not been widely demonstrated. Reasons for limited efficacy might include phenotype, stability, expansion, or specificity issues. Polyclonal Tregs might not be specific enough or might not expand sufficiently after infusion. Polyclonal cells hang around for about a month but then rapidly decrease in the blood. Antigen specificity. Antigen-specific Tregs are far more potent than polyclonal Tregs. In the NOD mouse model, a quarter of the number of antigen-specific Tregs was more effective at curing diabetes than a large dose of polyclonal Tregs. Generating antigen-specific Tregs from humans using patient APCs pulsed with peptides has been attempted, but the precursor frequency in peripheral blood is often too low to get a usable product. Mouse models like NOD mice are clonal, making antigen identification and targeting easier than in genetically diverse humans. Engineered Tregs. Genetic engineer, genetic engine, genetic en- to address issues with polyclonal Tregs and the difficulty of expanding endogenous antigen-specific Tregs, generating designer Tregs through genetic manipulation is being explored. Genetic engineering can be used to imbue cells with desired antigen specificity, stabilize FOXP3, and add other characteristics. This can involve TCR gene transfer or CAR design. Engineered Tregs are also called CAR Tregs when using a chimeric antigen receptor. Engineering antigen specificity by TCR gene transfer. Involves transferring a specific T-cell receptor gene into Tregs. Often uses TCRs isolated from effector clones specific to target antigens, e.g., islet-specific TCRs from T1D patients. Lentiviral transduction can be used to express the TCR. Adenovirus vectors can be used to express FOXP3 to help reprogram effector cells into Tregs. TCR-engineered Tregs can suppress effector cells recognizing the same antigen or different antigens via bystander suppression in a site-specific manner. Islet-specific, but not polyclonal, ENG Tregs prevented diabetes onset in mice. TCR challenges for human translation include HLA. Challenges for human translation include HLA restriction. Each TCR is specific to an antigen presented by a particular HLA type, limiting applicability to certain patient populations, mispairing of introduced TCR chains with endogenous TCR chains, potentially leading to off-target effects, and ensuring function at the site of tissue damage. CRISPR-Cas9 can potentially be used to knock out the endogenous TCR when introducing a new one to improve pairing. TCRs are a more natural way to redirect specificity and work via the endogenous CD3 complex. They can potentially work in both the lymph node and the target tissue as HLA is expressed in both. Donor specificity can be conferred by transduction with CAR. CARs are chimeric antigen receptors. They typically use a single-chain antibody, scFv, to recognize a target molecule on the cell surface, linked to signaling domains, e.g., CD28-CD3 zeta. Second-generation CARs with CD3 zeta and CD28 signaling domains are commonly used in Treg therapy. CAR design allows recognition of surface molecules in their native conformation, independent of HLA presentation. A 2-CAR Treg specific for HLA-A2, a common MHC class I molecule, protected from skin pathology and prolonged vascularized mouse heterotopic heart transplant survival more efficiently than polyclonal Tregs in humanized mouse models. CAR Tregs show increased localization at the target site, e.g., human skin graft expressing HLA-A2, compared to polyclonal Tregs. The CAR helps the Tregs home and stay at the target site because they are seeing their target. Challenges for CAR Tregs include that they typically target molecules highly expressed on the target cell surface. While they work well at the target site, they may not work as well in the draining lymph nodes where effector cells see processed peptide fragments presented by HLA. An attempt to generate insulin-specific CAR Tregs for type 1 diabetes using a CAR against insulin, a molecule found in high concentration around beta cells, showed that while the cells expanded and survived, they did not protect from disease. This highlights challenges with CAR Treg stability and potential overstimulation. People are working on next-gen Tregs that address phenotype, stability, expansion, and specificity issues. This includes optimizing suppressive function, ensuring stability, e.g., adding a second FOXP3 copy, knocking out instability molecules, improving tissue homing, persistence, and metabolic reprogramming. Additional modules are being added, including for visualization in vivo. The future of Treg cell-based therapy. Advancing Treg cell therapy. The future of regulatory T-cell, Treg, therapy lies in developing universal donor cells that can be widely substituting the natural T-cell receptor, TCR, with a synthetic one boosts the specificity of these cells, while also preventing potential mismatches between receptor chains. Efforts are also underway to develop advancing Treg cell therapy. The future of regulatory T-cell, Treg, therapy lies in developing universal donor cells that can be widely applied in adoptive immunotherapy. Cutting-edge tools like genome editing and the ability to derive T-cells from induced pluripotent stem, iPS, cells are paving the way for Treg cells that are compatible across all major histocompatible complex, MHC, types. These engineered cells can be customized with synthetic immunoreceptors to target specific antigens based on individual patient requirements. Treg cells may be harvested from healthy donors, via peripheral or umbilical cord blood, or generated from iPS cells created from fibroblasts. After expanding these cells in the lab, precise genome editing, often using CRISPR-Cas9, is employed to enhance their function by adding synthetic receptors and tailored components like an orthogonal Illinois-2 receptor system to boost survival. By removing native HLA genes and inserting non-classical variants, the modified cells can avoid immune rejection and natural killer, NK, cell attacks. Substituting the natural T-cell receptor, TCR, with a synthetic one boosts the specificity of these cells, while also preventing potential mismatches between receptor chains. Additional enhancements may include the introduction of chemokine receptors for targeted migration, expression of suppressive cytokines to strengthen their regulatory function, and built-in safety switches to terminate the cells in case of unforeseen side effects. Technologies like the SynNotch system allow precise control over when and where these engineered traits are activated. Efforts are also underway to develop universal receptor platforms that allow easy adjustment of antigen specificity. Innovations such as convertible CARs, Xiphos Biosciences, ACTRs, Unum Therapeutics, and switchable CARs, Calibre, separate the recognition and signaling components of the receptor, making it possible to fine-tune the targeting ability of Treg cells. While originally designed for effector T cells, these systems could be adapted for regulatory T cells to direct them to inflamed tissues more precisely. Additionally, novel TCR-based technologies like the TRUC platform from TCR2 Therapeutics merge antibody fragments with TCR subunits, enabling antigen recognition without HLA restriction while engaging the full TCR signaling complex. Translation and challenges. Multiple opportunities exist for Treg cell therapy in human disease. Antigen-specific FOXP3+ Tregs are an attractive option. Stability, homing, and persistence issues need to be addressed for engineered Tregs. Safety and feasibility have been shown in multiple human trials for polyclonal Tregs, but efficacy has not been widely demonstrated. Clinical trial design is challenging for cell therapy. Doing properly placebo-controlled trials is difficult, especially regarding the logistics and ethics of control groups receiving apheresis but no product. Generating designer Tregs involves processes like lentiviral or AAV vector transduction. Generating GMP-grade viruses is a significant cost. The overall process is expensive, involving specialist environments, pure reagents, and takes 14 to 28 days. Isolation alone can cost around £10,000 due to the purity of antibodies. The cost is currently high, £0.25 to £0.5 million per dose, but may decrease as more people enter the field and processes scale up. There is interest in developing off-the-shelf products, potentially using Tregs generated from induced pluripotent stem cells, to avoid deriving cells from each patient and create large cell banks. Big Pharma is investing in this area. Academic research has led to the founding of companies in this field, such as Quell Therapeutics, a King's College London spin-out. Ongoing commercial trials include the Liberate trial with A2 CAR Tregs in liver transplant recipients and planned trials for T1D.